the way that we um, kind of operate with regard to food production and food sales, there's been some shifts that are making it easier for pathogens and tougher for epidemiologists to figure out what's going on. So not too long ago, a few decades ago, there used to be this trend of small grocery stores. Farmers in the local area would grow a lot of the food that you saw. And then, and often you knew your farmer, you knew who you were getting your food from. So you even knew a little bit about their farming practices. That has certainly changed. Now with the superstores that we have, you can get food products from all over the world, even here in the UP. And of course, you don't know how many of these other countries run their food production, what kind of food safety laws do they have in place, let alone how do they grow their food and what do they do to the food as it's growing in the fields. We've also created situations where there's a selection pressure on microbes to actually become more virulent. And primarily the big culprit for that is antibiotic use. And they have done numerous case studies where they can show, you give antibiotics to, let's say, cattle or pigs, and you can then sample their feces and you can show antibiotic resistance genes getting spread to higher population numbers. Then you take the antibiotic away and you stop giving the pig the antibiotics. And then you sample over time again, and you can see that the antibiotic genes, the frequency of those goes down. And so you've removed that selection pressure, you've removed antibiotic use. And so, yeah, definitely antibiotics play a selection pressure. It works the same in human populations too. If we keep giving people antibiotics when they don't really need it, it's just out there more in the population. You get more and more of the bacteria or fungi becoming resistant. Take away the antibiotic, take away that selection pressure, and you see a drop in resistance. And pathogens become more sensitive. So it's not rocket science, but if you don't understand selection pressure and evolution and adaptation, it kind of gets lost in the shuffle and people don't really think about it. We've already talked about this numerous times, this idea that bacteria in particular are just really willing to share genes, including resistance to antibiotics. And that can make them more virulent. And also, you think about virulence factors that are on genes that can easily get transferred from bacteria to bacteria. So not only for resistance to antibiotics, but also their ability to be virulent, whether it's producing certain toxins, proteins, enzymes. So they're just really good at sharing all that. They're very collegial that way. Then you think about the human population and how our health has been declining over the decades. Think about all the immunocompromised people, including is dropping down into younger and younger populations, not just the elderly or really sick people. Certainly those with cancer and or autoimmune issues, more serious diseases, long-term chronic disease like aging baby boomers and other populations. The older you get, the less your immune system works well for you, starts to decline. And even something as simple as this, the use of antacid drugs like Prilosec, which if you watch the ads at all, I still see plenty of ads for Prilosec if I ever bother to turn the TV on. And that is creating a situation in your gastrointestinal tract that allows for pathogens to survive your stomach, because it's altering the pH of your stomach. And then if you happen to ingest some pathogens, food or water or some other drink, and then they can make it through your stomach that much easier. So again, that doesn't sound like such a big deal. Well, I'm just taking some antacids. What's the big deal? But it can actually, long-term chronic use of that can really make it a little dicier for you. I like this quote. Animals are living in medieval conditions and we're living in the 21st century. Consumers have to be aware that even though they bought their food from a lovely modern deli bar or salad bar, it started out in the 1600s. So he's referring to the feedlots that are currently used here in America and elsewhere in order to jam, in this case, cattle into feedlots, vast acreage. They're all hanging out together. Mostly this isn't on grass. This is all pretty much dirt, and they're getting fed all kinds of questionable material. And so part of the process of doing the more modern way of raising meat is to give antibiotics. They just routinely do that, even if they're not showing signs of disease or infection. I think I mentioned this before. They figured out that if you give low doses of antibiotics to cattle and pigs and so on, they put on meat quicker. It tends to be slightly better quality meat as well in terms of the marbling and what's desired for humans to eat. And so they'll do it even though there's not infection. It gets your animal to market quicker, and you can usually make a higher profit. Other countries are starting to wake up and say, probably should not do that. And there's certain countries overseas that have outlawed it, but we have not. And so the bulk of antibiotics are actually into the animal production. It's not given to humans for direct human diseases. It's actually to deal with. And then, of course, in feedlots, you do have a higher rate of pathogen spread because of their close proximity to each other. So there's 19 different classes of antibiotics, and only 7 are given to humans. And again, 70% of all antibiotics are going to animals for food production. So let me give you an example. That's kind of a classic case study. And this just makes me feel so sorry for epidemiologists in today's day and age. It's such a challenge. If you like hard mysteries to solve, become an epidemiologist. All right, E. coli 0157H7. This case study was in hamburger meat. And what the epidemiologists, of course, wanted to do is find out where did this pathogen come from? What's the initial point source, reservoir? What can we do to try to stop this? So they found initially that, when you think about that last picture I showed you, those cattle and feedlots, okay, so think about that. And then they're sent to centralized slaughterhouses. So animals from different feedlots can all converge into one slaughterhouse and get processed there. And then they can go to the plants for cutting it up and getting it packaged and all of that. And then when they get to the step where they're making the hamburger, they combine meat from many different bins. As they're doing that, there's selection process really. And just keep in mind too, as we follow through this story, that when you make hamburger meat, you're putting it through a grinding process to make like little spaghetti strings of meat, right? And that increases the surface area. Not only that, but if you've ever bought a hamburger that hasn't been made into patties yet, you have to open the package, you have to grab a chunk, and then you have to manipulate it, right, in order to make the patty. Well, as you're doing that, some of the stuff that's on the outside gets mixed to the inside as you're making your little ball, and then you flatten it out. All right, so you have to think about this idea that things that might be growing on the outside of that meat might end up internally more in the center of the patty. So think about that. And then, okay, so if all of that is going on, and then eventually it gets to the grocery stores or restaurants, they found that one hamburger patty may combine meat of 100 different animals from 4 different countries. So this is done through DNA sequencing. where they're able to actually do this kind of tracking in this level of detail. But if you're an epidemiologist, can you imagine trying to figure this out? Where you have deadly E. coli infecting people and killing them, and you're in a race to figure out where did this start. And then you have this kind of food processing system in place. So this case study with Jack in the Box, which was, I believe, out in California, when this happened, or where it happened, when they traced it all down, 443 different cattle they could trace the patty to. So it's all mixed up into one patty from 6 different states, from 5 different slaughterhouses. How do you track that? Probably not even born when this outbreak occurred, I think. It happened quite a while ago, but it was one of the first that where we had the technology to really track down all the sources of the meat. And this was kind of an eye-opening experience to see the data roll out from this. And so this is making the job very difficult to figure out what, where to go to stop it. Unfortunately, this particular case study did cause some deaths of young children. And you have to think about body size in this case. E. coli, when you have a much smaller body mass individual, it's much easier to overwhelm the system. And plus, their immune system wasn't fully built yet. And so there were some deaths with this particular outbreak. So E. coli is nasty, this particular one. Okay, now we have billions of E. coli in us right now, and they're fine. It's actually a good thing. There's many, many good E. coli. But this one, the 0157H7, they've now renamed it to Shiga toxin E. coli, and STEC is the abbreviation. And so the O and the H are referring to the antigens. If you take immunology, I'm sure he would, Dr. Sharp will talk to you more about this. But basically, it allows this E. coli, this particular strain, produces enterotoxin, which I'll talk about in more detail. But it's called a virotoxin. That's what but it's a type of enterotoxin. And so the Shiga is also called the Shiga toxin. And of all the toxins that we are aware of that bacteria produces, is the third most deadly. So I have in the notes here, Shiga toxins ravage the cells lining the intestines. The bleeding permits the toxins to stream into the circulatory system. Toxins tear apart red blood cells and platelets, which can lead to brain hemorrhaging, uncontrolled bleeding. Clots can form in the bloodstream, blocking tiny blood vessels around the kidneys, heart, and brain. As the kidneys give out, the body swells with excess waste fluids. All major organ systems can be affected, leading to stroke, blindness, paralysis, heart failure. So incredibly potent toxin and very much a systemic effect on the whole body. So again, if you know that this is in hamburger meat and then trying to track it down, and you are in a race against time, because this is something that can kill, and it's not anything to be taken lightly. So E. coli also, it doesn't just pass through, it will set up camp in your intestines. So it's able to linger in your intestines and keep pumping out this toxin. And so E. coli tends to end up contaminating meat, especially ground meat. And there's been numerous case studies over the years. But it can also be spread through water, either drinking water or swimming water. There's been cases of it showing up in swimming beaches, for example, coming from fecal matter of someone who's infected, or there's been implication of like waterfowl, their feces, causing the E. coli to get into swimming areas where humans are swimming. So this one is definitely a concern. Then there's this idea. So let's say you do get, you eat something and you get sick from it. And then you survive it. You're like, okay, I'm all better. Done with that. And most of the time that's true. Most of the time your immune system fully recovers, your body fully recovers, but there are a few cases where it's like, well, it could come back in other ways to haunt you. So salmonella can, for example, even after you have recovered from the salmonella infection, you can get reactive arthritis getting triggered later in life. You can get Guillain-Barré syndrome from Campylobacter jejuni, which is a neurological disorder. And again, that can happen many years after your infection. The E. coli, you can get permanent kidney damage if you survive it, where it's just going to be a problem and you might need a kidney transplant. The fancy word or phrase for that is hemolytic uremic syndrome. And then I already mentioned Listeria monocytogenes. This one, if you survive it, that's great, but it can cause, especially if you're pregnant, cause miscarriages. It can lead to meningitis even after a full recovery from the initial infection. And so basically, what they're saying is that some of these pathogens could trigger things later in life, including some autoimmune reactions, and basically something that you have to live with, a more chronic issue that you have to live with. So this is another reason, of course, where they want to have a good, clean supply chain of food and drink for us to be exposed to. So this is not a total list. There's other pathogens out there, but these are some more heavily studied where they can document the connection between things happening a little bit later in your life back to certain infections. So there's certain pathogens that the CDC is actively tracking, monitoring. And this table gives many examples, give you some of the ideas of what it's doing. So one thing I'll mention again, but I wanted to point out here, norovirus. Look what most common cause of diarrhea, right? So people often think when they get the stomach flu, oh, I have the stomach flu. They think it might be a bacterial, like salmonella or something, and it's probably a virus that was in that water or food that you ate that made you feel so sick. It's probably a virus. And so that's a lot of people don't realize. Most of the time, though, you recover from that. No problem. It's just not fun. I'm sure you've heard like cruise ships. We've had a series of cruise ships that had issues with norovirus. So that is definitely a problem, still is a problem. But then you see Salmonella, Campylobacter, E. coli, Clostridium. So I'm not going to be asking details on these to this level of detail that they're showing on the table. It's more for your own knowledge and hopefully trying to prevent yourself from getting sick. But I would like you to recognize the names of these pathogens, all right? So I think it's a good idea to be aware of these. And I think it's Shigella. I'm pretty sure it's Shigella. That I was looking to see if they had it in here, but they don't really say. Shigella is interesting because at least the case studies I was reading, 10 cells is all it takes to get sick. You get sick from 10. That's not very much. That's pretty impressive. So you don't need a high dose for that one for sure. And you've probably heard about Toxoplasma gondii, that parasite that can make you change your behavior. You heard about this one? It does it with mice. When mice get infected, then they don't get they're no longer fearful of cats. And they think it's toxoplasma is kind of controlling the thought process. It's able to alter your brain function so that you'll be more, what's the word? Less fearful, but more not courageous, but take risk taking. There we go, risk taking. And they've actually done like controlled studies where they infect mice or compared to non-infected and then present cats and watch what's going on. And the infected ones are like, oh, hi, want to be friends? And the cat can eat them. So then that's good for toxoplasma because it has to get back into the cat to finish the life cycle. So they've even shown some interesting data with humans. When humans are infected with toxoplasma, it also increases risk-taking behavior. So just be aware of that they're collecting some interesting data. So the way to eliminate that is generally don't play in sandboxes when you don't know what's been in that sandbox. And I think about all the years when I was a little girl, I played in sandboxes all the time. And then if you have a cat, and cats are awesome, if you have a cat, just be careful when you clean the litter box. And one thing that I certainly loved when I was pregnant, I got off. I never had to clean the cat box the whole time I was pregnant. It was awesome. So that's an added benefit because you don't want to get any exposure to it for pregnant, as it can cause problems for the developing fetus. Okay. What else? So we've been looking at pathogens, the ones that can make you sick, and in food or drink, and thinking about the idea that you can't see them, you can't smell them, you can't taste them. And then you have the other group of organisms that spoil food, which in general you can, oh, yuck, the food or drink is nasty, smells bad, looks bad. Basically, when we're thinking about the pathogens that make you sick, they have very specific genes that on their chromosome or their plasmids that allow them to present or to manufacture these virulence factors and toxins. And remember, they can get spread through horizontal gene transfer. So they're often referred to as islands. So they call them pathogenicity islands in this case, because they're clustered together on the chromosome or the plasmid, and then they're easily spread as a section like that because they're all in the same spot. That spot can get easily spread under microbes. And so that was a pretty decent explanation for how easily pathogens were acquiring these traits of becoming pathogenic, because some microbes, there's some well, I think it's a little bit later, because there's some microbes like Clostridium. No, sorry, I'm really tired. Diphtheria. Why am I blanking on the species that causes diphtheria? Anyways, that one, you can be exposed to that one just fine. But if it got infected through transduction mechanisms by a virus that carried the genes that allowed for it to become virulent, then you're in trouble. So it's not always the case that it would harm you. So basically, we're looking at the most serious cases seem to be pathogens that acquire genes that allow for toxin production. Then they can be very potent. And it doesn't really matter. They can be found in food and drink. Clostridium botulinum is the most deadly toxin. Botulism toxin is the most, that we're aware of, the most deadly toxin produced by a living entity. And the way that it works is it causes what they call flaccid paralysis. So paralyzes your muscles in a flaccid state. If babies ingest honey that has Clostridium botulinum in it, and they start to get the symptoms, they actually call it floppy baby syndrome, which is kind of a sad term, but that's actually what they call it because the baby can't tense muscles at all. And so if you see the baby going floppy, that's rush to the hospital because you have to appreciate your diaphragm is a muscle. And so if that is paralyzed in flaccid state, you can't take in a breath, you can't exhale a breath, you can't breathe. And so that's the danger, is that your diaphragm goes paralyzed. So here you can see in that image on the left that the endospores, when they're inside the rod shape of Clostridium, they have a very characteristic position in the rod. They're usually just, they're not at the end, they're just subterminal, they call it. And so it looks like a club, like a billy club type idea. And that's very characteristic. So if they saw that in the clinic under the microscope, they should immediately assume Clostridium. I have in the notes that you can boil the toxin. If you boil it, it can destroy it. So that's why you may have heard, you know, if you open a can of soup, make sure you boil it before you eat it, especially if the can was bulging. Don't ever eat anything with a can that's bulging, by the way. I would not recommend it. It's kind of Russian roulette. So the idea here is heating or boiling, but it's a risk. Okay, I'm going to give you a couple of ideas of other toxins. So staphylococcus food poisoning. So I bolded or made a point. So when we say poisoning, if your doctor says you had food poisoning, that has a specific meaning. Basically, that means that the bacteria produced a toxin that your body responded to. And again, and you can't really taste it or smell it. You don't know. Why it's a problem is that, so let's say you go out to eat and you eat eat a hamburger or something. The toxins, if Staph aureus is on the meat, it's producing toxins as it's on the meat, growing, doing its thing. And so you can get exposed immediately to toxins right away, as soon as you swallow that food or drink whatever staph was in. It's a type of enterotoxin. But unfortunately, this one, enterotoxins, can be challenging because they're relatively resistant. So if you dry out things or heat things, it doesn't really destroy the toxin. And even boiling doesn't destroy it. So because you can immediately get exposed to toxins as soon as you ingest that food, it's usually a quick onset because they start acting pretty much as soon as they get in your body. And so 1 to 6 hours after you eat, you may have had this already happen in your life where right around in that time frame, you're like, oh man, I don't feel good. And that could be what you should be thinking about. It could be something like this. So this is the poisoning idea. And poisoning usually means if you're getting exposed to preformed toxins, they're already there in the food. And so it's a quick onset. So these, let me go through the enterotoxin idea. So basically, when you look at toxins, what we're aware of is there's 2 main categories and then like a subcategory. There's these so-called exotoxins. These are proteins. That's when we say they're exotoxins, they're proteins. Nothing else. They're protein. And certain bacteria make them. They're, from the bacterial perspective, they're doing this to be able to break open the host cell in tissues. And basically they want the nutrients to leak out of cells and tissues so that they can get access to them. So a subcategory of exotoxins are enterotoxins. So enterotoxins are exotoxins. They're given a special subcategory because they work on the small intestine. So entero is referring to intestine. So enterotoxins are exotoxins. It's just that they are being produced by bacteria hanging out in your small intestine. So this often causes, when they're doing their thing and producing these toxins in the small intestine, then what happens to your body is you start to release lots of fluid into your small intestine, and that leads to diarrhea and vomiting. I'm hoping the answer is no, but if you've ever been sick where you rush to the bathroom and you can't decide, should I kneel or should I sit? Because you don't know, it's probably coming out both ends, right? That's like the worst when you have to vomit and you have diarrhea. And so think about this. Hopefully that won't happen to you, but it's probably an enterotoxin that you're getting exposed to. Okay. The other idea is endotoxins. That's the other big category. Endotoxins are actually the lipopolysaccharide. So when we've already talked about the lipopolysaccharide when we looked at human microbe interactions. And so the LPS, that is part of the gram-negative bacteria. It's part of their cell wall. It's that outer membrane that intaken, it's part of the cell. And that outer membrane, has that outer leaflet that looks very different than a typical membrane, including having that lipid A portion. That lipid A is what our body most strongly reacts to. And so basically it can cause shock and death, potentially, because basically the LPS starts to overstimulate the immune system. And like I said, it can lead to shock. And we know from the human microbe idea that too much LPS exposure can lead to chronic inflammation of the body, systemic body inflammation. So this table is a good one to study. I'll be asking questions off of this, looking at compare and contrast exotoxins versus endotoxins. So you can see with the endotoxin, it's only gram-negative because we're zeroing in on lipid A portion of the LPS. And you can see that boiling doesn't work with the endotoxin. And then you can even, if you're running a fever, that can also help further figure out what might be going on. You can see the toxicity factor with the exotoxin. It can be quite high. Endotoxins. So even though you can't boil it to kill it, at least it has low toxicity. So chances are you'll survive it anyways. Won't be fun, but you can survive it. On the other hand, with exotoxins, it's a little bit more dicey. Depends what you're sick with. So this is going back to the idea that you can have certain bacterial exposure and you might be fine. But if they happen to acquire that stretch of genes that allows them to produce those toxins, then you're in for some trouble. And so often what they see is it's bacteriophages or phages that are transferring through the transduction mechanism. And so we're going back to that idea of the bacteriophage infects the bacteria, pastes its DNA into the bacterial DNA, and then the bacteria has the ability to express that stretch of DNA. And so that's the lysogenic idea, when the phage DNA that it brought over is pasted into the bacterial host DNA. And so if it just sits in the DNA like that, then the bacteria can keep expressing it. And if it has toxin genes for toxin production, it will just keep producing the toxins under the lysogenic state. So they know that diphtheria, tetanus, botulism, cholera, this is all, they can track that it's a phage that infected those strains of the different bacteria that allows them to become pretty deadly. Okay, so toxin production is pretty serious. It can kill you. It can be in food and drink. The other big idea, besides poisoning, food poisoning, is this idea of foodborne infection. Foodborne infection. This is the idea that you eat contaminated food. The bacteria get usually to your gastrointestinal tract and set up camp. And then as they live their life, they start producing virulence factors and other things that'll make you sick. So it's an infection. You have to wait for the bacteria to actually set up camp and start living in your body, and then you start feeling the symptoms. So Campylobacter is famous for that. It will get into your intestinal tract. It attaches into the epithelium and can actually get in and start to inflame the cells and tissues. You generally will spike a pretty big fever and just feel awful. Headache, nausea, cramps, watery, and so it's really shredding out your intestinal cells there. But it's because it's setting up camp and living there and continually like making these different virulence factors. I got this website, found this off a website. 2/3 of packaged chicken contains salmonella or Campylobacter. That's kind of a disturbing thought. So, careful. Of course, it's planted. So you'd have to do, I'd have to look into that more carefully because the website I got it from, they're promoting vegan. So, but chicken is a big one. I've read other sources too where more issues pop up with infected or contaminated chicken than other meats, at least lately in the near, in the recent past year. Salmonella is another idea of food infection besides Campylobacter. And so again, you have to ingest it. It has to get into your intestines, survive, set up camp, and start living. This one, your doctor may say, oh, you have gastroenteritis, or they may say enterocolitis. That means typically when they say that, it's usually referring to the salmonella. And the most common culprit is Salmonella enteritidis spiravar typhimurium. And typhoid Mary idea is what we're talking about here. So this one is a facultative anaerobe. It's gram-negative, and it's actually related to E. coli and Shigella. You can find it in a lot of different stuff, eggs, milk. What happens with the eggs, by the way, is that the mother hen is infected. And then as she's producing eggs, it gets transferred to the eggs during the formation of the egg. So that's how the eggs get infected. So even if like you rinse off the surface of the egg, doesn't necessarily matter. It's inside the egg, so you can't get to it. Salmonella is pretty interesting because you can actually get into your phagocytes, which are part of your immune system. And phagocytes, their job is to engulf and ingest foreign entities and destroy them. But salmonella just invades it and lives in it. And so the phagocyte has this living salmonella inside of it. So they call it an intracellular pathogen, which makes it very challenging for your immune system. Your immune system has really no idea that salmonella is there when it's hidden away in one of your own cells. That makes it very, very challenging for your immune system. Because it's an infection, it takes a little bit longer for you to figure out something's wrong. So instead of 1 to 6 hours, like with a toxin, it's more like 8 to 48. And then you start feeling that headache, chills, vomiting, diarrhea. And usually a low-grade fever, usually not a real high one. You use self-limiting means you usually survive it, but you can be carriers like typhoid Mary. Hopefully not, but it can happen. So the idea that it can progress to typhoid fever, which is a systemic infection, so that if salmonella doesn't stay in your intestinal tract and instead goes systemic, you can get typhoid fever. That one definitely will spike a big fever. So Typhoid Mary is the classic example. You've heard about her in lab, and that was quite the case study for typhoid. But looking at all the bacteria, it's really viruses that win in sheer number of cases. All the cases every year, when they add it all up, time and time again, viruses win. Historically, that's what we tend to see. So foodborne infections, mostly it's viruses. So again, your doctor may say gastroenteritis, and that is usually they're thinking about, yeah, you got a virus. Nausea, vomiting, people say, oh, I was sick for 24 hours, but then I felt better. So it's usually a virus that's doing that. And then that's that off that table, the Norwalk or noroviruses. But there's others, rotavirus, astrovirus, hepatitis are all major players. This is not from your book, but again, I like how they chunk it out into bacteria and then protists and then the viruses. And with the bacteria, there you see B. cereus, rice, starchy foods, high sugar foods. But if you scan all the way down, viruses, 9,200,000 per year. And let's see, when it comes to the bacteria, I guess it's Campylobacter that wins, 1,900,000. Ooh, Campylobacter is way up there. And then total viruses, 9 million, compared to total bacteria, 4 million. So approximately double the amount of virus. So I like that table. It's a little bit more explicit, showing that it's, yeah, the viruses. Usually that's what's making you lay down on the cold floor of the bathroom and just wait for the next round of whatever is going to hit you. So what can we do about that? They have been trying to tackle this issue. How do we prevent food poisoning, food infection? And certainly with the idea of refrigeration and freezers, that was a huge jump forward from previous tactics. So certainly using cold, freezing, refrigeration, pickling your foods. Usually when they say pickling, they mean that they've added acetic acid. That's usually what's going on. Usually salting it pretty heavy or sugaring it. Depends on the food. They'll either use sugar or salt. So pickles, fish, peppers, and fruits are made this way. So when you buy pickles off the shelf at a grocery store, unless it explicitly says on the label that it's fermented with live microbes, this is what they did. They just took, they preserved it with acetic acid. So again, acidity is a good strategy. Pathogens that cause us trouble are adapted to our pH of our body, which is about neutral. So if you can create foods or drinks that have acidic pH, they don't survive that so well. If you can dry out food so that any free water is gone or very low water activity. So if you can, like smoked fish, a lot of the smoking and the smoked fish process is drying the fish plus smoking it. And that's kind of a double whammy to get rid of microbes that might be on the fish, for example. Heating certainly can help. It's not 100% necessarily. Good techniques in terms of when they are doing all the food prep. Chemical preservation is a big one. And once they figured out that certain chemicals can be used and spiked into food, but it's come under scrutiny and criticism because some chemicals not so good for our body either. They kill microbes, but they also can be a problem for us. And so they are starting to phase out some of them. And so be aware of that. Usually on the ingredient list, it's toward the end of the ingredients. So you have to be careful about that. Nitrates, a lot of nitrates, sodium nitrates and sodium nitrites. Those are chemical preservatives, but they're starting to be implicated in mutating our DNA. So be careful. Irradiated food and drink is a thing. And then fermentation. Fermentation is a great way to preserve your food. When I go out, if I'm going to be out hiking all day and I don't have access to a refrigerator and I know I'm going to get hungry, one of my favorite things to take with is a yogurt because I don't care that it gets to room temperature for hours on end. I don't have to worry about it because it's a fermented food. The chances of anything in there causing me trouble is minuscule because there's billions of beneficial microbes in there ready to outcompete. So fermented foods, again, my plug for fermented foods. I would hope that you give it some thought as you live your life, and because it has a lot of benefits in addition to preserving food. It also blends additions. Their waste products are good for us. Sounds kind of weird, but they can be very useful and helpful for our health. So think about that. And then also you ingest the microbes. That can be very beneficial if they set up camp in you. They can be long-term partners for you. These I'm just put in for your own use. I'm not going to ask questions on these tables here. This is just to get you thinking about if you wanted to get into fermentation, all the options that you might try. Okay, so this is just some ideas. And then one last thing. Don't forget about going to the Center for Disease Control and Prevention. So we call them the CDC, but they're actually, that's their full name. They have a lot of information, up-to-date information on food outbreaks or things that need to be recalled, or don't eat this, don't drink that because we have an issue going on. So even if you are traveling within the United States, this is always a good place to go visit and see what they got going on because they have really current data. So don't forget about that resource. Okay, we'll stop there and we'll start industrial microbiology on Monday.